Nice to hear of good opportunities out there. All right, well, finally figured out what the problem with the quizzes was, and it looks like I'm going to have to remake a couple of those. Um, Use a program called Respondus to upload them, and for some reason, files were getting corrupted in the upload, but got to figure it out. So 7 and 8 I was able to save those, and they are available to you. The next 4, though, 9, 10, 11, 12, I'm in the process of remaking those. I'm going to make them a little bit shorter so that we can get through them pretty quickly. And I'm actually just going to kind of leave it open for now. Tentatively, we're going to try to finish all those by next Wednesday, but we'll just kind of watch your all's progress and see if we need to extend those dates, then that's possible. Because that was my error. If you guys notice that, just let me know as soon as possible. I have one student that let me know that she could not view them, and so that was how I knew to go back in there and make sure that they were available. But you guys got to keep me accountable too. So anyway, we're on to chapter 13, talking about mineral nutrition and really how plants actually absorb nutrients from the soil and utilize them. So we'll move on to that. All right, so it's a little bit counterintuitive. It's not exactly what you would expect. We know that nutrients are able to enter water in the soil solution in their ionic form. However, they do not flow directly into the plant with that water that is absorbed, kind of as you would expect. I mean, often we use water-soluble fertilizers. However, there are barriers to the uptake of the nutrients themselves. And these barriers require specific systems that can be unique for particular nutrients to actually take them from the soil solution into the roots and move them throughout the plant. So most of these systems do have a strong attraction for a single nutrient and kind of are geared for particular nutrients, especially with nitrogen and potassium. There are other systems, though, that can move multiple nutrients using kind of the same mechanism. Of course, all plant nutrients are found in the ionic form of the elements, or in some cases in the molecules. So they are charged, and they can be cations, positively charged ionic forms of the elements or molecules, or they can be anions, the negatively charged. And some of these ions actually can have different charges in the different chemical forms. So as we can see, if we look down here in nitrogen, we've got ammonium over here on the far right, NH4 plus, it's a cation, but then nitrogen in the nitrate form or nitrite form is negatively charged. So that's going to vary the different ways that they're taken into the plant and how more efficiently they are utilized within the plant. So we talked a little bit about cation exchange capacity. That's the more important form of exchange of ions in the soil, but anion exchange capacity is actually important to look at as well for these negatively charged chemical forms. Of course, in the soil solution, the ions themselves are hydrated. That's actually how they're bonded to water, surrounded by these water molecules. So the plants take up the water, but then they have the barrier for the nutrients themselves. So how are they actually absorbed? Well, there's a few different ways, and of course, once they are inside of the plant, they are transported by the xylem and phloem tissues. And for a long time, it was thought that there was just one, they're unidirectional, but now we know that they are both multidirectional. But generally, xylem mostly is moving water and in some cases ions as well upwards to shoots. Phloem is mostly moving photosynthates down from leaves to other plant parts as well as lateral movement. Both xylem and phloem, though, can go down and up. So here's just a cross-section of a root, and we can kind of see down here the meristematic zone. We got this region where there's rapid cell division and differentiation. We got the elongation zone. This is like the root cap right here. The elongation zone is right behind that meristematic zone, and then the maturation zone is actually where most of these nutrients are actually taken up through the root hairs rather than through the root tips. So we're going to see these different lines showing our different uptake through the phloem and through the xylem. Notice that if we look at the phloem, it actually extends further than the xylem down through the growing root, because it has to transport nutrients from the leaves to the roots to be utilized in that meristematic zone. Whereas the xylem is more, I mean, it's moving water downwards as well, but it's more moving it upwards through the plant. Also notice that we have this endodermis here, and in the endodermis there's what we know as this Casparian strip. And the Casparian strip is basically just this barrier. It's a waxy deposit in the cell walls that prevent movement of both water and ions into the xylem, unless they are taken up and moved through the Casparian strip through various means. We'll get to. First, think about the plant cell walls. Cell walls are composed of the carbohydrate cellulose and hemicellulose as well as pectin. But the cell wall, even though it provides rigidity to the cell, it's actually very porous. And spaces in between these cellulose fibers and pectin can allow unimpeded flow of ions and water into the root. So we see this here as apoplastic movement, and basically this is just the movement of water and ions, mostly ions though, through the cell walls. And you can kind of see here it's taken up like along the root tip and then through the cell walls, and then into the xylem and phloem. And we have in the endodermis, where that Casparian strip is, we have cells with walls that are coated with this waxy substance known as suberin. And in this case, it creates an impenetrable barrier for both water and ions, and they have to pass through the plasma membrane or plasma lamella in order to actually enter into the root and be able to be transported. So this different route is called the symplastic route, and that's actually moving through the cytoplasm. And of course, it's a little bit different. So apoplastic movement here through the cell walls, because they're very porous, is quicker, but not all nutrients can be taken up apoplastically. Symplastically, as it takes a little bit longer, but some nutrients can only be taken up symplastically. So two different types of movement from nutrients in the soil and the soil solution into the root tips, either through the cell walls apoplastically or through the endodermis and through the cytoplasm being symplastic. So we have basically this, sorry, moved a little too fast there. the plasma membrane, which we talked about earlier on, we're looking at photosynthesis and the movement of energy, composed of this kind of lipid bilayer, which is impermeable to ions alone. But the ions have to get through the plasma membrane in order to be able to be utilized in plant tissues. So how do the ions get through the plasma membrane? Well, we actually saw this image before when we were looking at the plasma membrane. It's through these proteins that are embedded in the membrane, those that are extrinsic kind of on the outside versus those are intrinsic on the inside. And these serve as channels or carriers for these ions. So basically, it's kind of similar to just movement of water. The carrier proteins or movement of the photosynthates basically take up through their various charge sites. You can see here negative and positively charged sites on the proteins. They're able to kind of absorb the ion and then move it through the plasma membrane. Pretty cool. And one very important thing to note in terms of plant nutrition is that elements can act synergistically with each other, meaning that they are able to be absorbed together and they can act together, work together basically. However, some elements can be antagonistic to each other and sort of repel each other. And it's really not necessarily just the amount of nutrients we have in a soil and in a soil solution and thus in the plants themselves that we are concerned about. It's the balance of nutrients. So just a couple of examples here. Potassium uptake, for instance, can be enhanced by adding calcium to the soil. So that's a synergistic effect. Calcium and potassium work together. However, phosphorus can interfere with the uptake of iron by a plant. So we can have both deficiencies and imbalances of nutrients in a soil solution, and then thus also in a plant, which can cause, it will look like nutrient deficiencies often, but it's not necessarily just a deficiency, it can be just an imbalance. So we can have an overabundance of particular nutrients as well. So we have 17 essential elements, and some texts look at these differently and include more as well. You should know what the essential elements are. An element is considered essential if a plant has to have it in order to complete some part of its life cycle, whether that's just the growth of a particular tissue or the actual fruiting, or maybe it needs it for seed production. It doesn't necessarily mean that you have to have the element in order to get a viable crop product necessarily, although usually you do. But if you're growing a vegetative crop, like lettuce, for instance, you don't necessarily have to have all 17 essential elements because you're harvesting it before it completes its life cycle. So 17 essential elements for plants to complete their life cycles. And these are divided into particular areas. We've got our major macronutrients over here on the left: carbon, hydrogen, and oxygen, which these CHO, remember these are parts of sugars, carbohydrates, glucose, sucrose, all of that is primarily composed of carbon, hydrogen, and oxygen. These make up the bulk of plant tissues. However, these are not really so much mineral nutrients. We do get some of these carbohydrates from the soil, but more often we get carbon, hydrogen, oxygen from carbon dioxide gas and from water. So the CHOs are extremely important, but we don't generally add fertilizers that supply these. We add water, or we add organic matter in some cases can supply these carbohydrates, these hydrocarbons, I should say, carbon, hydrogen, and oxygen. The others, though, these other major macronutrients we supply in an elemental form in the soil, or sometimes through foliar application. We get phosphorus, P, potassium, K, nitrogen, N, and sulfur, S. These are our major macronutrients. And if you, sorry, I accidentally clicked there. If you actually take a plant and dry it out completely and did a molecular analysis of what is left in the plant, 99% of the plant is going to be in this, column right here. We have major micronutrients as well. So these micronutrients are needed in larger amounts: calcium, Ca, iron, Fe, magnesium, Mg, boron, B, manganese, Mn, copper, Cu, and zinc, Zn. So those are micronutrients that are needed in larger amounts. And then the last three over here, molybdenum, nickel, and chlorine, are really just needed in very small amounts, Mo, Ni, and Cl. And some plants don't need them at all. So they're kind of more like facilitative nutrients. Some plants do need all of these. Some plants do not need all. So it's on a per plant, per crop basis. And there's a little mnemonic device here that is used to try to remember all of these essential elements. C, Hopkins Cafe, managed by my cousins, Mo, Nigel, and Clyde. So you can make up your own there, but if you look at just the elements here, C being carbon, Hopkins being the rest of the major macronutrients, Cafe being calcium and iron, managed, Mg, by boron, my cousins, Mn being my cousins being copper and zinc, Mo, molybdenum, nickel, Nigel, Clyde, Cl. So just remember that you do need to memorize these essential elements. Know what plants need in order to complete the life cycle. As I mentioned there earlier, more than 95% of plant dry weight is composed of just carbon, hydrogen, and oxygen. And that bumps up to 99% if we include the NPK and S. So again, these are coming from mostly from water and gas. Carbon dioxide and water is where we get most of our CHOs. The remaining elements are mineral nutrients primarily coming from the soil. Again, we got the primary macronutrients, secondary macronutrients, and the micronutrients, also referred to as trace elements. And this table is just kind of showing you what each of those nutrients are used for in the plant. And we'll talk about some of these, but not all of these today. If you look down here, especially at the trace micronutrients like molybdenum, nickel, and zinc, especially on zinc here, legumes need zinc for seed production. These are not required in every single plant. And I mean, it's such a small amount that typically soils are not devoid in these elements. However, if you're growing particular crops, you want to know which micronutrients you do need. So if any element is deficient, any of these essential elements, then a plant will not be able to complete its life cycle properly. And it may not be able to grow and develop it properly. So Justus von Liebig, an early plant scientist, developed what he calls called the law of the minimum, which we still look at today, which basically states that one element that is found in the least amount deficient is going to be the limiting factor for growth and development in a plant. So you have to have all elements in sufficient quantities in order to be able to actually allow crops to produce in most cases. And the best way to really to look at this is through soil testing. Whether that be a test kit that you pick up or if you send it off to Virginia State University or Virginia Tech to be tested at their soil labs. We also have to look at the mobility, not just in the plant, but also in the soil as an important factor in plant nutrition. In some cases, mobile elements can be moved within the plant. So, for instance, nitrogen, very mobile nutrient, can be utilized from older leaves and moved to newer leaves in a plant. What the book kind of refers to as scavenging these elements. Can't do that with immobile elements. And that's actually really an important thing to look at in terms of nutrient deficiencies. If an element is mobile, then you'll see a deficiency in the older leaves. If it's immobile, then you'll see the deficiency in the younger leaves. So let's first talk about these major macronutrients. First, nitrogen, which is the most commonly deficient nutrient in agriculture. It is so important because nitrogen is found in every protein. It's a primary element in these carrier proteins, which move other nutrients, as well as DNA itself. The two main forms of nitrogen that plants can absorb and utilize are ammonium and nitrate, NH4, the cation plus, and nitrate, NO3 minus, the anion. So unavailable forms exist, like nitrite, for instance, in the soil, and they are made available to plants through the nitrogen cycle, which is just similar to the carbon cycle, critical to life on Earth, but it basically is just a series of chemical reactions occurring in the atmosphere and in the soil, as well as within plants and other animals as well, in ecosystems. And as I mentioned earlier in the class, nitrogen is actually the most abundant gas in our atmosphere. And there are actually several ways to fix nitrogen into the soil. It can be fixed naturally by rhizobium in the roots of legumes and some other plants. It's just basically a bacteria that is able to take it and put it into a mineral form into the soil. It can also naturally be fixed by lightning. So lightning actually inundates nitrogen into the soil as well. You can kind of see it. Of course, it can come from other sources. It can come from mineralization of, say, like manure, from animal waste, or from the death of plant and animal matter, mineralized into, in this case, ammonium, and then nitrified and converted from ammonium to nitrite and then to nitrate. And then it can also be denitrified. That would be nitrate being broken up into nitrite and then nitrogen gas. That's denitrification. So this is an extremely important cycle here. Nitrogen is also, it's very mobile within the plant. It's also very mobile in the soil too, which is another reason why it's often a limiting factor in agricultural production. So in order for plants to be able to use nitrogen, they can't just take it out of the atmosphere. They have to have something to be able to do that for them. So we have nitrogen fixation, again, by particular types of bacteria. The most important of those being rhizobia. which are not just one species, actually several different genera and many different species. They live in the nodules and the roots of legumes like peas, beans, soybeans, alfalfa, and also other leguminous trees too, like locusts, for instance, and some other plants. It's not just the legumes that are able to fix nitrogen. Some plants in the rose family are also able to fix nitrogen. Some nitrogen-fixing legumes are grown as cover crops and then plowed under or killed with herbicides to return nitrogen to the soil. And generally, most species of rhizobia only will infect one species of plant. Here's kind of what it looks like here. It's this sort of symbiotic relationship where the plant supplies the bacteria with the carbohydrates, the photosynthates really, and the bacteria supply the plant with ammonium. So it's a win-win situation. It's a mutualistic relationship here. And here's what it looks like in the nodules of the roots. And there are some cases in which, I mean, if you're growing legumes, that doesn't necessarily mean that you're going to have nitrogen fixation. Often we will actually inoculate the seeds with the bacteria. So you can actually purchase a rhizobium inoculant and then put that on the seeds and plant them. Generally, many soils, especially if they have not been fumigated, they have some, naturally occurring rhizobia in the soil. However, it's not a bad idea to use the inoculant. If you want to see, if you're noticing that you're growing, say, legumes, and they don't appear green, nitrogen is responsible for that kind of dark green growth. They don't appear green, then you might pull a plant up and see if you actually have nodules forming on the roots. If you don't, then you probably need to inoculate your seeds. So here we're just looking kind of at the process here where the rhizobia enters the root on these nodules, and that center picture there is a cross-section of a root nodule, looking at the different tissues of the infected region where that bacteria occurs, as well as the cortex of the root there and the vascular bundles. And then that last picture is just a electron microscope image of the interior of the cells housing millions of these bacteria. Of course, much of our nitrogen is today supplied by nitrogen fertilizers, and this is done through what is called the Haber-Bosch process, named after the people that first created it. They're taking atmospheric nitrogen and converting it to ammonia, then to ammonium, then to nitrate or nitrite, under very high temperatures and pressures. So they use methane as a source of energy, kind of as a catalyst, to use this high temperature and pressure to bind it often to a salt in order to get our nitrogen fertilizer. And it's very similar to what kind of similar, I should say, kind of a similar process as what the bacteria are doing, except this is really energy intensive and then quite expensive. And you may have noticed here that carbon dioxide is a byproduct. However, in the biological process, CO2 is still a byproduct here, but it's a little bit different. In this case, the energy that they get is in the form of sugar from the plant. So it's much more efficient and inexpensive than the high pressure, high energy form of the Haber-Bosch process. So bacteria are actually able to do this at room temperature and pressure. So it's interesting that we have, even with modern science, we still have not been able to equal nature in its ability to fix nitrogen. So we can often get nitrogen from sources like manures, other natural sources, compost, for instance. Worm compost is actually a really high nitrogen soil amendment. Or we can also get it from nitrogen fixing bacteria, things like cover crops, or we can get it from fertilizers. So nitrogen availability to plants depends on the transformation of the organic form of nitrogen to the mineral form. So the organic form, not talking about like certified organic, we're just talking about it in its natural state. The elemental form, nitrogen, or nitrogen gas form, N2, and then transforming that into the mineral forms of nitrate and ammonium. This begins with mineralization or ammonification, which is that conversion of that natural form of nitrogen to ammonium, and then converted to nitrate through nitrification. We can actually supply it to plants as ammonia, urea, ammonium, or nitrate. But if we supply it in the forms of ammonia or urea, it has to then be converted to ammonium in the soil before plants can actually take it up and use it. This is a very important part of the nitrogen cycle. So if we apply ammonia or urea, we have to basically rely on mineralization and ammonification occurring in the soil in order to convert it to a form that the plants can actually use. Nitrate is an anion, and being negatively charged, that makes it much more easily leached from the soil, because as water enters, it's positively charged. So it can bind to nitrate and leach it more easily. So that can happen naturally through precipitation or through over-irrigation, which can contribute to nitrogen deficiency, but also to pollution of things like waterways. If you look at water, you see algal growth in water, natural waterways. That is a significant sign of nitrogen runoff. And nitrogen runoff is actually the second highest pollutant of waterways, right after just soil sediment itself. When we look at nitrogen deficiency in plants, the first thing that we generally see is yellowing or chlorosis of the lower leaves. And that will continue to spread throughout the plant if not corrected. So generally, I mean, if you look at this tomato plant pictured here, you can see it looks kind of pale and sort of sickly on its own, not just the lower leaves, but overall the plant just, it does not have enough nitrogen. So nitrogen is a main part of the chlorophyll. I mean, there is at least one atom or molecule of nitrogen in the chlorophyll molecule that, so basically it's responsible for dark green growth. If you see light green or yellowing, that's often a nitrogen deficiency, and can easily and quickly be fixed because it's so mobile. And also being mobile within the plant, that's why you see it in the older leaves versus the younger leaves. Because basically when the plant needs more nitrogen in the new growth, it's going to take it from those lower leaves. Of course, in things like tomatoes and other crops, we have death of lower leaves is just a part of the development of the plant. You know, leaves aren't meant to stay alive forever. Once they've kind of done their job, then they naturally senesce. So don't always think of just death of leaves as just a nitrogen deficiency. You have to look at the whole plant when looking at deficiency symptoms. Phosphorus, of course, is another one of our major macronutrients, P, and it plays many vital roles in the plant's cells. First, it's present in the energy-rich compounds in these energy transport chains of ATP and NADPH. It's also part of the lipids that make up the membranes, known as phospholipids because they have phosphorus in them. And it's also a component of both DNA and RNA. So phosphorus is incredibly important and often looked at as the second most limiting element for plant growth. Generally in soil, it's often tied up in organic matter. And many of the phosphorus compounds are only slightly soluble. It's a much less mobile nutrient than like nitrogen, for instance. Another thing that we have to look at is the soil's pH for the ability for plants to actually uptake phosphorus. And generally, between 6 and 8 is what we're looking at. So are really hovering around neutral is most beneficial for uptake of phosphorus. We can have a fungi, a fungal relationship, a symbiotic relationship with mycorrhiza, which often forms this sort of fungal sheath on the outside of roots. It helps to uptake phosphorus and is often today used in many different commercial growing mixes. You can also just purchase mycorrhizal amendments to add to your soil as well to help with the uptake of phosphorus and other nutrients as well, but phosphorus is one of the big important ones. For instance, we actually here at VHCC, we use a mycorrhiza inundated form of our ProMix for our potting soil. And you can kind of see here that fungal sheath grows on the outside, but it actually can grow into the root and help to provide an uptake system for phosphorus, basically making it link from the organic matter where the phosphorus is bound into the cortex of the root itself. And here's just another kind of look at a different type of mycorrhizal fungus that rather than making a whole fungal sheath on that outside of the root, it actually enters the cells of the root cortex and can absorb phosphorus from the soil, also carry it back to the plant in exchange for carbohydrates. So again, it's another one of these mutualistic situations where the plant's supplying the photosynthates to the fungus is supplying phosphorus, not by fixing it, but by linking it to organic matter or mineral sources in the soil. And as I mentioned, mycorrhiza is available commercially. Phosphorus deficiencies often kind of look like a little bit more of a reddish or purplish look, especially on the undersides of leaves. It will also cause plants to kind of look a little leggy and kind of spindly. So in phosphorus deficiencies, you can still see dark green growth, but kind of a bronzing of the tissues and then a reddish or purplish color, especially on the undersides of the leaves. And you can see in this sample here, these plants are growing in straight perlite, which perlite is not really, does not have really any cation exchange capacity. So it's decent for starting things like cuttings, but it's not really good for actually growing plants. So as you can imagine, there's not really going to be phosphorus supplied in this situation. It's not going to be held even if it is going to be supplied. It will actually often just be leached off. So mycorrhiza is actually, there's many different species of mycorrhiza and some work better for different plants. So there are some cases now where there are commercial mycorrhiza that are actually particular for particular crops. However, often what many like commercial growing mixes use is just a bunch of different types of mycorrhiza in the mix. Potassium, K, is also critical. One of our other major macronutrients. It's critical to control the opening of stomata, the pores on the undersides of the leaves. It triggers both opening and closing of the stomata. So it's important for both photosynthesis, so it can allow it to release carbon dioxide, as well as transpiration, allowing it to release water vapor. It's also required for the activation of many different enzymes and proteins within the plant, and can have synergistic effects with other elements. Potassium is often retained in the soil because of its positive charge, so it's not easily leached. It's not incredibly mobile within plants either. Sources of potassium on the earth are actually quite plentiful and often in mineral sources. Usually potassium is not devoid in a new soil. If you're breaking new ground, typically potassium is there in a sufficient amount for the first few growing seasons until you export enough crop to remove that potassium from the soil. So it can be a limiting factor, but it's much less so than nitrogen or phosphorus. We get kind of similar deficiency symptoms of potassium as we do in nitrogen. We get chlorosis of leaves. It's a little bit different in dicots and monocots, though. In dicots, you'll see leaves starting to appear yellow and then some browning or necrosis of the tissues. It starts at the margin of the leaves and then goes inward. And you can kind of see here in this picture, although it's a little bit blurry, it's kind of an interveinal chlorosis in between the veins of the leaves. In monocots, it starts at the tip of the leaves and then spreads along the margins. And it can kind of look a little bit like a fertilizer burn in some ways. If uncorrected, it will involve the entire leaf as well as younger leaves of both broadleaf and monocot plants. Let's look at calcium. Calcium, of course, is also it's kind of a secondary macronutrient. Some people consider it a macronutrient. Some people consider it a micronutrient. It's kind of on the border, really. But it has several structural roles in the plant, including making pectin a stable structure in the cell walls, giving the cell walls rigidity. Think of calcium in plants kind of similar to what it does for people. It's used for our structure, right? Our bones. So deficiency can be a problem, not necessarily just if the plants or the calcium is not available in the soil, but if the plant cannot transport it to rapidly growing tissues fast enough, especially in things like fruit. So it results in the tissues collapsing and becoming soft and appearing rotted. And we can see this in things like tip burn in leaves of lettuce, or in tomato plants, it looks like blossom end rot, which often is confused with blight. Of course, blight is a fungal disease. Blossom end rot is just a calcium deficiency or an imbalance of calcium and magnesium. You can see blossom end rot. Again, it's on the flowering end of the fruit. Fruits, of course, are rapidly growing and developing tissues. And if they cannot get the calcium quick enough, then it will look like it's rotting. And it does look like blight. It's often confused with it. But just know that blossom end rot is a physiological condition and not a disease. They've also looked at it in an improper development of poinsettia bracts. Fortunately, it can be fairly quickly corrected with foliar applications of calcium. You can also supply it in the soil. And if you've probably heard old timers growing tomatoes using things like eggshells rich in calcium to prevent blossom end rot. Or also I've heard of people putting tums at the base of tomato plants in order to prevent it by supplying calcium. Of course, in that form, calcium carbonate. Here's a deficiency symptom of calcium in pansies, just deformed growth there. Kind of looks like the rotting of tissues. Magnesium also plays several different roles in plants, but the two that stand out is that one, it's an essential component of chlorophyll, and also it's a necessary component of ATP and the reactions involving energy transfer in plants with ATP. Generally, what we see in deficiency of magnesium is intraveinal chlorosis of the lower leaves, again, because it's a mobile nutrient. Because it's intraveinal, and if you, let's see if I can zoom in here. If you look at the leaves here on the tomato plant, notice it's in between the veins. The veins are still green themselves, and the zinnia back here, it's intraveinal chlorosis, yellowing in between the veins. That distinguishes it from nitrogen deficiency. And probably the most common treatment of magnesium deficiency is just to apply Epsom salts or magnesium sulfate to the plants. Another one, and I keep saying tomatoes because I've had a lot of experience in growing tomatoes as a cash crop, but we have kind of a tomato planting mix that we would use, and it would involve using things like eggshells and Epsom salts, in pre-plant application when you're planting the hole that you've dug. So that's to balance magnesium and calcium. They kind of have a synergistic effect, so you need them both in a balanced amount. Iron is another common deficiency symptom, and usually it's mostly due to pH. So you have to correct pH problems in order to get good iron uptake. It is an immobile nutrient, so you'll see intraveinal chlorosis again, except in this case it's of the upper foliage instead of the lower leaves. So that can be a good indicator for iron deficiency. And iron deficiency can be uptaken actually through the leaves, through foliar applications of iron sulfate or an iron chelate applied directly to the affected foliage. However, generally, one of the best ways to prevent or correct iron deficiencies, especially in the soil, is to correct the pH. Again, between 6 and 9, ideally hovering around 7. The neutral pH, the sweet soil, as many people call it, is important because it has the greatest balance of nutrient uptake for all nutrients. There's some nutrients that are better at higher pHs or lower pHs, but for the greatest balance of all essential elements, it's right around neutral. So whenever you're diagnosing plant problems, just looking at the typical deficiency symptoms can be misleading. So you need to look at the whole plant. You also have to look at the symptoms from plant to plant. And in our crop projects, we'll be looking at some of these deficiency symptoms. So you guys will actually be teaching each other about some of these symptoms in different crops. Also, you have to look at it because it could be more than a single element because of the synergistic or antagonistic effects that some elements have on each other. And many of these deficiency symptoms can also look like plant disease or other disorders. So you need to recognize what is disease, what disease looks like, what are the key diseases of your crops versus what are the deficiency symptoms. One strategy to kind of determine whether it's a deficiency or a physiological condition or a disease is to actually just apply a small amount of your suspected nutrient in question to a small area to see whether that corrects it or not. And then if that deficiency is confirmed, then you continue your application. You can also, of course, do soil testing. Good idea to do that before putting your crops out. Also, you can do tissue analysis. There are tools out there that can look at especially the macronutrients by analyzing leaf tissue. Also, you can send these tissues off to laboratories. Because reproduction is so important to the plant's life cycle, plants will actually allocate resources to the most important parts for them, which is often the fruits and seeds. And that process is known as remobilization, taking those nutrients from other plant parts and moving them to the reproductive parts. And here's just remobilization kind of depicted in a graph in beans. So you can see they start to reduce it and move it to the actual seeds themselves. And that's it for Unit 2 in our chapter on mineral nutrition. So try to get as many of the quizzes done as quickly as you can. I'm going to try to make them all available today. So hopefully by the time you guys get out of class, they'll all be back up again. Most of these, from 9, 10, 11, and 12, and the one that I'm making for 13 will be like 5 questions each, just to give you guys a little bit of extra time. And I'll be watching you guys and grading them as you go. So just try to get on those quizzes as soon as possible. But do you have any questions or anything? All right. Well, you guys have a great day. Great weekend. We'll see you next week.
Okay. Bye.